students. Today we'll be beginning up with a very interesting topic and a basic topic in biology which we call as the cell. Cell, the unit of life. As simple as that, it is a basic structural and functional unit of our lives. Now in this topic, what all things we will be covering? First of all, we will be studying something about the plant cell. We will be studying something about the animal cells, and we will be also studying something about the bacterial cell. So all these three cells we will be studying in detail. Also, we will be seeing their components. Now to begin up with this thing, cell. You must be wondering from where does this word come cell from. So let's begin our journey with the cell. The first scientist who found out or who coined the term cell was none other than scientist Robert Hooke. He was the scientist who gave us the word cell. Now to begin with, what he did was, he took up a bark of a tree, he isolated certain cells and he visualized those cells under the microscope. At that time, the microscopes were also very simple microscope. So what he observed under the microscope was just empty spaces because they were the dead cells. When he observed the cells, it reminded him of the monasteries where the monks live. And at that time, the monasteries, they were called as cell. And this is how he gave us the word cell. So now you come to know who gave us the word cell. Yes, it is the scientist Robert Hooke. Now we come to forward to study one more great scientist. The name of the scientist is Antoine van Leeuwenhoek. Antoine van Leeuwenhoek, he was the first person to study living cells. Robert Hooke also studied, but those were the dead cells. This person, Antoine van Leeuwenhoek, he studied living cells. And at that time, when he studied these cells, this microscope which he used was a very simple microscope, just like a magnifying glass. After that, it is said that this person has built around 400 microscopes. You can imagine 400 microscopes were built by him. Also, he made various discoveries regarding the living cells. This was about the microscope. The commonly used microscope which we have today, this microscope is called as the compound microscope. This compound microscope can give us a magnification of up to 400 times. So, you can get a magnification of up to 400 times by using this compound microscope. The scientists did not stop over here. They went further and they built a more complicated, a more sophisticated microscope which we call as the electron microscope. So if you just see this electron microscope, it is a very huge microscope. Over here in this electron microscope, it can give us a magnification of up to 2 lakh times. So you can imagine how huge magnification power it has. So by using this electron microscope, the scientists, they were able to explore so many things regarding the cell, regarding various structures which were available. Now we come forward to study about two great scientists. The name of the first scientist is Sheridan. He was a botanist. What he studied was, he took up various plant cells. He studied all these plant cells and he came to one conclusion. The conclusion was, all plants are made up of a structural and functional unit which we call as cell. Similarly, there was one more scientist called as Theodor Schwann. So this person, Theodor Schwann, he was a zoologist. Similar discoveries were made by him. He studied various animal cells and he came to one conclusion, that all animal cells or all animals are made up of one structural and functional unit which we call as the cell. Years later came the third scientist. His name is Rudolf Virchow. This scientist, he made a postulate saying all cells which are present have come from pre-existing cells. So we have three scientists coming up. These three scientists, what they said, they gave us three statements. Years later, these three statements are now known as the cell theory. Now we will see the 3 points which were given by the three great scientists Rudolf Virchow, Matthias Sheridan and Theodor Schwann. Let's have a look at it. The first point says about the cell theory. The first point of cell theory says that all cells are the structural unit of life. The second point says that all cells are the functional unit of life. And the last point states that all cells arise from pre-existing cells. So these are the 3 points. The first point says that all cells are the structural unit of life. Second point says that all cells are the functional unit of life. And the third point says that all cells arise from pre-existing cells. Now let us go forward to understand these 3 points. The first point says all cells, they are the structural unit. What do you mean by structural unit? So if you just take up any tissue or if you take up any organ of our body, if you make the smallest part will be of a cell. So that is forming the structural unit of life. The next point says that all cells are the functional unit of life. Now, functional means what? The all cells which are present in the body, they will all do some or the other function. So that forms the second point, which says all cells are the functional unit of life. The third point says all cells arise from pre-existing cells. What does this point mean? This point means that whichever cell has come into existence, it is because of some earlier cell. So by the process of cell division, these cells have come into existence. This is all we will be studying in the cell theory part. Hello students. Now the next part which we will be seeing today is the examples of a few cells. So let's begin with the first cell, which is the smallest cell. So we have the smallest cell, which is a bacterial cell. It is being said that the size of a bacterial cell is just within a few micrometers. So this is the smallest cell which we have to study. Next we have is the biggest cell or the largest cell. The largest cell which has ever existed is the ostrich egg. The largest cell which ever has existed is the ostrich egg. It is being said that this ostrich egg has a diameter of about 18 centimeters. So you can imagine how big is this one single cell. Also, we have one of the longest cell in our body. The longest cell in our body is that of a neuron. Yes, neuron is right. What is neuron? Neuron, it is nothing but a cell which is helping to conduct all impulses from our body. So this is the example of smallest cell, largest cell and the longest cell. Now scientists, they wanted to classify cells. So they have classified cells on the basis of its cell size, its cell number. So this classification which I have written over here is based on the cell size. So this classification is based on cell size. Now let's move forward to study the classification which is based on cell number. So on the basis of cell number, we have 3 categories of cell. First is unicellular organisms. So yes, you have guessed it right. The unicellular organisms, the example is again a bacterial cell. We also have certain algal cells which are again unicell. So bacterial cell is one example. The second category which we have is few celled organisms. Means these organisms just have a few cells. So few celled organisms. One such example of a few celled organism which we have is Spirogyra. So if you see Spirogyra, this is one cell which is just having a few of these cell organelles. The next category which we have is multicell. Yes, so multicellular organisms. Can you think of some examples? Yes, one major example is humans. Among the plants, you can take example of a mango plant and various other cells. Now this was about how the scientists classified cells on the basis of its cell size and on the basis of its cell number. Now you might be thinking that what is the benefit of a cell being very small? So first benefit is that if a cell is small, it can communicate well with other cells. So first benefit of a cell being small is it can communicate well with other cells. The second benefit is that its mass by volume ratio will also decrease. So when the mass by volume ratio decreases, it can easily spread up things, it can easily exchange things with its environment. So these are the two benefits why a cell needs to be small in size. Also, if you see, there are various shapes of a cell. If you observe our RBC cell, so RBC is nothing but the red blood cell. This red blood cell is having a different shape. You see the melanocyte cell which secrete the pigment melanin. It has a different shape. You observe a plant cell. Even in a plant cell, its root hairs will be different. Its flower cells will be different. So, what am I trying to say is, based on the function and based on its work, the cell will be of different shapes. So, if you pick up any cell from our body or from any plant or from any bacteria, they will have their different shapes. If you take up amoeba, so if you just have a look at amoeba, it is irregular shape, which is not having any well-defined shape. So, it is because of its function, the various shapes of cell exist. Students, on the basis of the cell's nucleus, the scientists have divided the cells into two types. The first type we call as prokaryotic cell and the second type is called as eukaryotic cell. Now these are two different words. Let's have a look at it. The word pro means primitive and the word karyo means nucleus. So over here it means that these cells have a primitive type of nucleus which is an old type of nucleus. Now let's have a look at this eukaryotic. What do you mean by this? The word eu means true and the word karyo means nucleus. So it shows that these organisms have a true type of nucleus. Now just have a look at it. The prokaryotic cell and a eukaryotic cell. So if you just see, the prokaryotic cell is not having a well-defined nucleus. Means the nucleus is not enclosed in a proper nuclear membrane. And if you have a look at a eukaryotic cell, the eukaryotic cell is having a well-defined nucleus. Now let's list down a few examples of the same. So, example of a prokaryotic cell is bacterial cell. We also have one more example which we call as cyanobacteria. Now these cyanobacteria, these are blue-green cells. Now let's come over here, over at the eukaryotic cells. So let's take a few examples of a eukaryotic cell. So eukaryotic cell, the first example is a plant cell. So this is how a plant cell looks. The second example which we have is of animal cell. So see, this is how an animal cell looks. And the third example which we have is of a fungal cell. So see, this is how a fungal cell looks. Hello students, today we will be studying about a very famous cell which we call as the bacterial cell. We will be studying its components in detail, but before we begin with a bacterial cell or with a prokaryotic cell, let's have a look at its components. First of all, a prokaryotic cell will not have a well-defined nucleus. So if you just see the diagram what I have drawn over here, this diagram is not having a proper nucleus. It is just a nuclear material which I have drawn. Also, this prokaryotic cell is a very primitive type of cell which I have already mentioned. A prokaryotic cell will have four major things. First is its cell wall. Second is its cell membrane. Third is ribosomes. And the fourth thing, which is the most important thing, is the nuclear material. So if you just see, let's have a look at all these one by one. So the first component of a prokaryotic cell which we have is the cell wall. Now the cell wall of a bacterial cell is made up of peptidoglycan. This is a very complex type of a material. So the cell wall is made up of peptidoglycan. Now let's talk something about this peptidoglycan. Peptidoglycan layer is a very complex layer which gives a tough covering to this bacterial cell. Now this layer is a freely permeable layer. The word peptido comes from the word proteins and glycan comes from the word carbohydrate. So this layer is a mixture of peptides and carbohydrates, that is proteins and carbohydrates. So if you see, this is the layer, peptidoglycan layer. This is a very thick layer. Now one more important thing, on the basis of this peptidoglycan content, whatever is the content of this peptidoglycan, scientists have divided these bacteria into further two types. The first type we call as gram-positive bacteria and the second type is gram-negative bacteria. Now the classification which I have given you just now, it means that these gram-positive organisms have a very thick layer of peptidoglycan and gram-negative organisms will have a thin layer of peptidoglycan. This was one reason why this classification is done. Also there is one second reason, that the staining method which is used was devised by the scientist Gram, and the organisms which took up the stain, they were called as gram-positive, and the organisms which did not take up the stain, they were called as gram-negative organisms. So I hope the concept of cell wall is clear with all. So cell wall in a bacterial cell is made up of peptidoglycan. This peptidoglycan is a mixture of peptides and glucose, or peptides and carbohydrates. On the basis of peptidoglycan content, scientists have divided organisms into two types. First is gram-positive, second is gram-negative. Gram-positive and gram-negative I have already explained. Now let's move one step inside. So if you just have a look, inside the cell wall we have nothing but the cell membrane. Cell membrane is a layer which is partially permeable, or you can also say this layer is a semi-permeable layer. What do you mean by the word semi-permeable? Semi-permeable means it will allow entry and exit of only a few molecules. So that is the reason this layer is also a very protective layer for the bacterial cell. It will allow entry and exit of, say, limited things like water, salts, sugar molecules. So only few things can enter and few things can exit from this layer. Now this layer is not made up of peptidoglycan. but this layer is made up of proteins and lipids. This layer is also called as lipid bilayer membrane. So this is what is called as the two layers of the bacterial cell, which is the cell wall and the cell membrane. Inner to the cell membrane, we have a fluid-like matrix. This matrix is called as the cytoplasm. So if you just have a look, this is the cytoplasm. So this matrix, fluid matrix, is cytoplasm. Cytoplasm is the place which will hold all the cell parts. The cell parts like the ribosome, nuclear material, plasmid, these all together collectively we call them as cell organelles. Cell organelles, or in a better way we can call them as organs of the cell. So cytoplasm is the place where all cell organelles are embedded. What is the nature of this cytoplasm? So the cytoplasm is basically made up of proteins. It will have various cell organelles embedded in it, and it is the place where all cellular metabolism will take place, or all cellular chemical reactions will take place. So this is the place. Now we will study certain parts of a bacterial cell. So if you see, the first part is the ribosome. Now ribosome in a bacteria is of 70S type. So ribosome in a bacteria is of 70S type. What do you mean by this S? S is nothing but the Swedberg unit. S is nothing but the Swedberg unit. Now this 70S, it means that while the scientists, they were finding out the content of ribosomes, they used centrifugation techniques. So when they did the centrifugation techniques, one of its layer, it got settled in the 70S subunit, and hence the name is given as 70S subunit of ribosome. But if you further study this ribosome of a bacterial cell, it is made up of two further units. The first unit is called the bigger unit, and the second unit is the smaller unit. So the bigger unit is made up of 50S, and the second unit is made up of 30S. Now that doesn't mean you add 50S and 30S. It is completely different. So these are the two subunits. If you just have a look, the 50S subunit and the 30S subunit. This is how the two subunits look. What is the main function of ribosome? Yes, the main function of ribosome is to synthesize proteins for this bacterial cell. As we require proteins, the bacteria also requires protein. So this ribosome is helpful in making various proteins for the bacterial cell. Now let's move forward to study about the food granules, which is over here. These food granules, these are stored food materials by the bacterial cell. If the cell cannot consume a lot of nutrition, it will store it in the form of this granule. These granules again will be suspended in the cytoplasm. The next cell organelle which we have to see today is plasmid. So if you just have a look at the plasmid, this plasmid is the extra DNA material which the bacterial cell will have. This plasmid will give the bacterial cell special abilities, say like antibiotic resistance. It will also help the bacteria in something called as conjugation. So this is the reason plasmid is a very important part of a bacterial cell. If this plasmid is not there, the bacteria can survive. But if this plasmid is there, the bacteria will get an added advantage. Now let's come to the nuclear material of this bacteria. The bacterial nuclear material is definitely made up of DNA. DNA is deoxyribonucleic acid. This DNA is just in a single strand. So single strand means what? Just in one line. So the bacterial cell will have a single stranded DNA. This single stranded DNA is just coiled around and placed in the cytoplasm. And that is the reason we call a bacterial cell as a prokaryotic cell. I hope now you have understood why is it called as a prokaryotic cell. So this is how it looks. So these are the various parts of a bacterial cell, the main parts of a bacterial cell. Yes students, now we will move forward to study other parts of this bacterial cell. The next part which we have to study is a layer which is outside the cell membrane. That layer we call as Glycocalyx. calyx layer. Now this glycocalyx layer is again of two types. First type of glycocalyx layer, we call it as the capsule layer, which surrounds the bacterial cell outside the cell wall. What is the function of this capsule? This capsule gives an added advantage to this bacterial cell, and it will prevent this bacterial cell from sticking to any substance. Say, be it an organism's WBC. If the WBC, white blood cell, will come and try to engulf this bacteria, capsule will help this bacteria to escape. Another type of glycocalyx which we have is the slimy layer. The slimy layer is also present in some bacteria, which is same way outside the cell wall. Now this slimy layer also, what it does is, it will not allow any bacteria to stick to any substance, and it can also be a virulent factor. What do you mean by a virulent factor? Virulent factor means the ability of that bacteria to cause disease to another organism. So the virulent factor to the bacteria is given by the slime layer, and capsule layer will give overall protection to this bacterial cell. Also, there are other parts in the bacterial cell which we call as, the first part is the cell extension part. The cell extensions which we have to study is of two types. First is the flagella, and the next one which we have is the pilus, or we also call it as pili. Flagella, if you have a look at the diagram of flagella, so if you just see this flagella is coming from the cytoplasm. So if I have to define this flagella, I will say that the flagella is a cytoplasmic extension. So if you just have a look from the cytoplasm, it comes from the cell membrane and cell wall, it pierces out and comes out in the outer part. So this flagella, the shape is like a whip. So the shape is whip-like. Also this flagella helps the bacteria to move forward. So this is helping in the movement of the bacteria. Another thing which is also one cell extension which we call as the pilus. Pilus is a singular form, pili is plural. Pilus also same way provides the locomotion to the bacteria. The bacteria can move very well. There are various type of pilus which are present, or various type of pili which are present on a bacterial cell. But one such pili which we have to study is called as the F pilus. What is the function of this F pilus? This F pilus helps the bacteria to transfer its genetic material to another bacteria. So F pilus is also called as fertility pilus, and the process of transferring genetic material means this, the nuclear material, from one bacteria to another bacteria, the process is called as conjugation process. So who helps in conjugation process? It is the F pilus which will help in conjugation process. Along with that, there is also one more thing which is present in this bacterial cell which we call as mesosomes. So this is what is called as a mesosome. Mesosomes, if I have to define it, they are invaginations of the cell membrane. What do you mean by invaginations? Invagination means when this cell membrane will come inside, or it will fold in a depression, we call that as an invagination. So these invaginations which are present in this cell membrane, what do? They help the bacterial cell in various functions like DNA replication, to secrete out something. This is how the mesosome helps the bacteria.